appreciate that. And Neil's reminded me of the talk I gave at Parkinson's Queensland, which must have been 10 years ago. 20 years ago.
We were there. Yeah.
Oh, you were there, so. And the science has progressed hugely since then, and a lot of the concepts that I can't remember what I talked about, but the concepts I was talking about, I think now are being in the process of being delivered, including the plans that I had for my research so long ago. So today I'm going to be talking about stem cells. Everyone, stem cells are such of interest in the community and in the newspapers, anything to do with stem cells. And I want to talk you through what stem cells are and the kinds of things that we're trying to do with stem cells, and specifically for Parkinson's disease, and show you where we're going, and perhaps to give you a caveat about all of those other stem cell transplantations that are out there at the end of Dr. Google, which are unproven, but we'll come to that later. So you probably all understand that statement that Parkinson's disease is a combination, like so many of our diseases, of some genetic susceptibility, some genetic risks, and some environmental risks. So there's barely a disease in which if you have a gene mutation, you will get the disease. We read in the newspaper, we now have the gene for disease X. Well, that's not true. Even disease X, which has such a strong genetic component, has lots of other factors in it. And in Parkinson's disease, there are rare variants, rare mutations shown up there on the top left there. You don't need to know the details, but these rare combinations of mutation, or these rare mutations, travel in families, so they're identified in families where you can see in the generation there's this gene mutation that's passing down. And those are high-risk genes, and then there are others which are identified as being low risk. That means if you and your family had that particular gene that was being passed around, it might give you a 10% increase in the risk of Parkinson's rather than a 90% risk. So when you hear about gene mutations in Parkinson's disease, there's a great variety and a great variety of risks associated with them. And the reason that the lower risk, the more common variants, the more common mutations, the reason it's a lower risk is because there are a number of other gene mutations which will be involved. There will be age-related changes that are involved. There will be environmental exposures that will be involved, and we'll talk about some of those later. So genes, for most of us, genes do not determine outcomes. They contribute to the outcomes. But it's true that everybody who has Parkinson's disease has a set of gene mutations that are going to make them susceptible. And if they get Parkinson's disease, we know they've got those susceptibility genes. And part of our search in science, in Parkinson's and neuroscience, is to find those genes and find how they contribute to changes in the cell functions that lead to Parkinson's disease. So what do we currently do with Parkinson's disease? Well, as you know, we treat symptoms. We look at, here's a brain that's affected by all of these changes, and it leads to motor symptoms, and they're the ones that we first identify, and they're the ones we first treat, and they're the ones that your DBS is treating and your L-DOPA drugs are treating. And that's what cell transplantation currently is aiming to treat. We know, and I'll come to that, but we know the cells that are responsible for those motor symptoms. And the idea is if we can treat those, replace those cells, then you can treat that major symptom. But of course, as you know, that's only part of the problem. It's a debilitating part of the problem, but you know that there are lots of other effects which are not going to be treated by sticking cells in one part of the brain, are they? So the other use of stem cells will be to find drugs. And of course, drugs can go to every part of the body, and they could treat at a deeper level, at a biological level, an understanding of the biology of the disease through looking at cells and then trying to find a drug which treats that biology. And that's really what we've been doing in Parkinson's disease for some years, and I'll be talking about that. And when you do a Google search for stem cells, you come up with this whole alphabet, this whole mishmash of words, mishmash of different kinds of stem cells, embryonic stem cells, induced pluripotent stem cells, mesenchymal stem cells, which is what many of these bogus treatments, they suck out a bit of fat, they spin out mesenchymal stem cells, and they stick them back in you in the host. Okay. Oh, I see. Oh, yeah, that's much louder. Is that better, Nod, or put your hand up? Great. Okay, thank you. So there's a whole mishmash of words on stem cells, about the different kinds of stem cells, and I'll tell you a bit about those first up, just to get you in the picture. So the first stem cells probably that you became aware of are embryonic stem cells. Those were the ones that brought all of the legislation in the early 2000s. And they had been explored in other creatures up until a publication in 1998 when they were first, somebody said, yes, we know how to do this in humans now. And if you remember Dolly the sheep, to get to Dolly the sheep, they had to make sheep stem cells. And then she was made by then taking the DNA from a healthy cell, an adult cell I should say, and sticking it into an embryonic stem cell, taking out the nucleus, that's where the DNA is, in the embryonic stem cell, and putting the adult skin DNA into the embryonic stem cell, and that caused the adult DNA to kind of reprogram itself to become an embryonic stem cell. They made Dolly the sheep. And so this is how you get an embryonic stem cell. So you fertilise an egg, and you wait for a few days, and you get a ball of cells, the blastocyst. And inside that ball of cells is this little clump of cells here, which this is about 7 days, I think. And the ring of cells around the outside go to make all of the supporting parts of the baby, the umbilical cord and the everything else to support the baby to implant it. It's only these few cells in the middle which go on to become the embryo. So if you then take that blastocyst and stick it in a womb, of course, you get a baby. But if you take that blastocyst and put it in a dish with a bit of a witch's brew, and this was, of course, where all the science came in, what was that witch's brew? But you wait, and you end up with a colony of cells, which we called embryonic stem cells. Now, just to give you an idea, this is about 100 times the magnification. So the cells in here are like these little dots you can see. Anyway, these clumps of cells are the embryonic stem cells. Now, of course, this has ethical implications for many people about embryos, but for scientists it was a Pandora's box. Well, no, better than a Pandora's box, because that has nasty connotations. But scientists could take these kinds of cells and really explore how they develop, how they get, how do you get this early development into a creature. Mostly it's done in other creatures, but early development in humans is how it's also explored. Then in the mid-2000s, only about 10 or 12 years ago, some Japanese people said, look, I explained how Dolly the sheep was made. You take the DNA from an adult cell and you stick it in an embryo cell without its DNA, and somehow it can do the stuff that an embryo DNA can do. So they said, well, it's got to be acting by something that's in the cell. It's reprogramming the DNA of the nucleus. And they did lots of their scholarly reading to identify some factors, and they went from about 100 different genetic factors down to 4. And so they took you can take any cell from, I've got a lovely baby picture, but any person. You add these factors, you wait, and you wait, and you get these embryonic stem cell-like cells from any adult and from any cell. People started off with the skin cell, that's pretty accessible, and then of course said, oh, what if we take some blood? Can we make it from blood cells? Yeah. What if we just take a scraping of the skin out of the cells that come out when you scrape inside the cheek? Yes, you can get them. What about if you get some of the cells that we urinate out because we shed them in our urine? You can make them from those as well. So I just think that's a bit shocking and a bit threatening in some ways. But anyway, you can make these cells from any cell type, any accessible cell type, which is important medically. Now, why are these cells important for us? Because if you take them and you transplant them into a mouse, like an immune-compromised mouse that can take human cells and not destroy them, they make all the tissues of the body. They're not organised like a body, but they come up a bit like a tumour. But this lump of cells, you'll see hair in there, you'll see teeth in there, you'll see muscle, you'll see heart. They just do this spontaneously if they're in a clump inside the mouse. But it proves that those cells could potentially make any cell of the body. And then the scientist's challenge is, well, if we take these in a dish, can we apply our ideas and our knowledge and our history to make the cell types we want to make to treat diseases? And both these embryonic stem cells and what we call induced pluripotent stem cells can do these things. I should say that the science of this is advanced. Now people are making, at least with mouse cells, they're making eye structures, they're making little brain structures, they're making little kidney structures, not just cell types, but they're actually making more complicated cellular structures, all, first of all, trying to understand the biology of it and also thinking about what the applications are for the future, medically. But we have other cells, other stem cells. These are the cells that we work on. And so stem cells have actually been around longer than since 1998, but most of us weren't aware of that. And they were the bone marrow stem cells. So bone marrow stem cells have been identified because bone marrow was used in cancer patients, bone marrow transplants, to restore their blood supply, their blood system after chemotherapy. So these are called hematopoietic stem cells. So there are cells there that their whole job is just to divide, and they give daughters which divide, and they give daughters which eventually become all of the different blood types that we've got, all of our white blood cells and our immune system and all our red blood cells. And we're doing this at the millions of cells a day rate. So those stem cells are very powerful little engines for dividing. And so they are used to repair the bone marrow after transplantation. But there are these other stem cells in the bone marrow that make a support for those hematopoietic stem cells, and they make bone, they make cartilage, they make fat cells, and they reside inside the bone marrow. And I mentioned mesenchymal stem cells. They are also in other kinds of tissue, and they're even in fat. And that's why the people who've got a liposuction tool for sucking out, your extra fat have now got onto this, and they said, well, we can suck out fat. And so they'll go around and say, well, if we suck out fat, we'll put them into anybody for almost any indication. Just because we can, and somebody will pay us $10,000 or $20,000. Unbeknownst to most of you, there are some parts of the brain which can also generate new cells, and they can make new nerve cells, and they can make new support cells called glial cells of the brain, and also the nose, as we'll come back to. So any repairing part of the body, when you cut yourself, there are stem cells, we call them adult stem cells, to differentiate them from the embryonic. That these adult stem cells sitting in your skin, and if you get damaged, their job is to divide, and their progeny will become the new skin cells. So, and you've got them in the gut because you swallow food, you're damaging your gut, you have to protect yourself. So, bone marrow transplants have been around for 50 years, and it wasn't really until during the 1980s that people started to identify how you got from a bone marrow transplant, and identified stem cells there that gave us, now we can purify those stem cells. I'm just saying that because they're stem cells which are now routinely used, as we all know, and very commonly used clinically. So in Parkinson's disease, again, as you've probably had explained, I'm sure, that here's a brain, and the cells that are responsible for your movement disorders are well down here, and at the base of the brain, in this place called the substantia nigra. And they send their connections up to the middle but higher part of the brain, where they release this drug, this neurotransmitter dopamine. And you can image that. You can image the dopamine levels in the brain and judge how biologically effective your transplant is. For example, if you're having a transplant, and some people, they started off with transplants of fetal tissue. So the Swedes, and then it was taken up in the US, so it takes, dissect open an aborted fetus brain and pull out these dopamine producing regions and transplant them into the brain. And when they published this work, the Swedes had more success than the Americans, which kind of just demonstrates how experimental these things are. And there are lots of reasons why one worked and one didn't, and it's still being done, I think, in Sweden. But a couple of things, and I'll talk about this in a minute, is that, of course, these people who had these said, look, we'll give our brains for you to analyse when we die. And when they died, they found that most of the transplants also had the pathology of somebody with Parkinson's disease. In other words, the cells they transplanted got the disease. So it's not a panacea. I mean, it gave them, it's a bit like DBS, it gave them a decent period of motor-free life, but it's not a simple biological problem by any means. And here's another example. These people were putting in these fetal tissue cells, and the question is, how many is enough? And that might vary between different people. And these people were putting 300,000 into people, and they found that they weren't enough. So you maybe have to put in more. So once you have something that seems to work, how do you fine-tune it? And this is part of that. You can see that the title says they got the dopamine back in this case into the right place, but it wasn't effective for symptoms. And this was a reference to a paper where they found the pathology in the cells that they transplanted. Okay, so let's get to Parkinson's disease and human trials and stem cell transplantation. So the idea is that you would get a donor that, like any transplantation, they're matched for their immune properties. You probably wouldn't want to get the cells from the person, because they've got those cells have the genetics that would definitely lead to Parkinson's disease. So then they could make these dopamine cell progenitors, that progenitor being the cell that's just about to become the mature cell that does the job. Or else you take embryonic stem cells and go this way. Or you could take these induced pluripotent stem cells. You could make the progenitors. You could put them back into the same person. Well, maybe they'd work for 40, 50, 60, 70 years like everyone, like they had. Or maybe you might have to re-engineer them so that they don't have the genetic risk. So this is the hope, and this has been the hope. As soon as embryonic stem cells were identified, Parkinson's disease became a target. So it's been, for 20 years that this dream has been around. And if you go to this website, clinicaltrials.gov, it's run by the National Institutes of Health in the US, and it's a place where all formal, serious clinical trials around the world are registered for everything. And if you type in Parkinson's and stem cells, you get a list of 12. And there's a whole lot of these, and these include fetal transplantations, they include embryonic stem cell derived neuron transplantations. It involves these iPS cell, it involves pig stem cell transplantations, all sorts, and it also includes some mesenchymal stem cell transplantations. So this is to say that it has reached clinical trials, that it has reached these first experimental trials in humans that stem cells and their derivatives are being used to transplant in Parkinson's disease. I'm not expecting you to read all of this stuff, but here's 4 sets of groups of, there in these columns, 4 sets in these columns of different groups who have all got together with, some are doing embryonic stem cells, some are doing foreign donor iPS cells, and some are doing autologous, that means derived from the same patient, put into the patient. But if you were to read this, you'll see that they've all got slightly different designs, because it's still experimental and they're still trying to work out which way to do it, and each of those groups has come up with slightly different ways of doing it. What's the best age to put them in? What's the best, how many years after diagnosis is the best time to put the cells in? How many cells do you put in? So once you've got something that works in your animal models, you come to humans and you've still got this experimenting done, and that gets done in this clinical trials period. Oops, wrong way. Okay, so that's transplantation work. Progress. Stem cells for drug discovery. How would you use stem cells in drug discovery? The thing about stem cells, you could take them from individual patients and healthy controls, and as a biologist, you could approach it as people in cancer research do. They'll take a healthy cell, and they'll take a cancer cell, and they'll compare their biology in lots of different ways, and they'll say, well, the cancer cells differ like this. Maybe we can find a drug to use that difference to kill the cell, right? And it won't kill the healthy cells. Our approach in brain disease is to say, well, can we find differences that are biological? Can we then find a drug that in a dish can treat the cell to make it back to normal function? And that could potentially become a drug for disease, and it's based on understanding the real biology of the differences rather than simply treating the symptoms, because we're looking at biological processes inside the cells. And after all of those genes I talked to you about, it's DNA that acts on the cell to change cell functions. And those cells, which in aggregate contribute or become our brain, and their differences in function is what gives us the disease. So studying the cells is important. So in this figure, it's saying you can identify chemicals at one end, and you go through a period of exploration of the biology shown in blue. That's where stem cells contribute, and then you go to the clinical trials phases on the right-hand side. So the stem cells we use, we get from the nose. They're accessible. ENT surgeon, I know some of you have had biopsies for our research in the past, and you take these bits of tissue out of the nose. Why the nose? Well, you can see here the olfactory mucosa, that's the tissue that gets covered in air and we breathe in. That's where the sensory cells are. And they have to grow their process, their nerve fibre, up to the red part there in the brain. That's where they're talking to the brain. And those cells get killed every day. They die every day by being in the nose. And I don't expect you to even think about understanding this particular figure. It's just got some things I wanted to illustrate. So imagine, it's just like the epithelium of your skin, but it has these sensory nerves which must go from the air that's up here, and they have a connection, direct connection with the air, and then they go down and they send their process to the nose. That means those cells are exposed to the air. And what's in the air apart from the odours? Some things are toxic, which could kill them, but there are bacteria, there are viruses. And we and others have shown that some bacteria can go straight into those cells, straight through and straight to the brain. So evolution has given us this great idea. Why don't we just, as soon as they might get infected with something, we let them die. But if you do that, you lose your sense of smell. And most creatures, less so with us, but most creatures, if they lost their smell like this today, how would they find their way home? How would they eat? How would they find their partner? How would they find know where their predator was? So it's it's great, we call it evolutionary pressure to do something about it, and that's to replace those cells continually. And in 1990, we discovered this stem cell, which sits down at the bottom of this thin piece of tissue, which gives rise to those nerve cells. And it gives rise to the other cells, the support cells, which in blue there at the bottom on the right, which help those nerve fibres get to the brain. And just as an aside, it's these support cells that we use in our spinal cord transplantation, for reasons I won't go into. But it's these stem cells which we can take from people. Now, they give rise to nerve cells and glial cells. Just like during development you have to have nerve cells make the same kinds of cells that make the brain. Here we have in the nose like a developing brain. And much of my scientific career was working out this mishmash of cells and all of the chemical signals which allow it to go from a stem cell to a sensory cell. We learned in about 2000 how to grow them in the dish. And so we began setting up our what we call neurobank, which we now have from biopsies that we've taken. We take this biopsy, we take it out of the nose, our ENT colleague does this, and we grow it in a dish. The great thing is, I told you that stem cells like to divide and divide and divide. That's their job. So we can keep them dividing and dividing and dividing in the lab. The other great thing is we can freeze them down and store them. So we can make up lots and lots of them. We can store them in liquid nitrogen at minus 179 degrees. And when we need to, when we want to do our experiments, we pull them out, we grow them up again. They're healthy, and we can do our experiments on them. So now, in our bank of 200 or 300 patients and healthy control cells, we've got about 50 for people with Parkinson's, and we've got matching controls, 50 or so. And our whole research program is based on the idea that we can take, for various brain disorders and diseases, and Parkinson's is one, schizophrenia is another, some genetic or highly genetic diseases like hereditary spastic paraplegia and developmental ataxia, telangiectasia, motor neuron disease. We take these biopsies, we do the cultures, and we can do a range of different kinds of biology. And this range of biologies has just exploded in my career. And the Human Genome Project, which we all know about, it's happened in the early 2000s, has been an absolute boon to this, and the technologies that we have available allow the kinds of things we now do with Parkinson's cells. But just to give you a quick insight to say that I reckon this process works, is this disease, hereditary spastic paraplegia. It's a highly genetic disease, and we chose patients with a particular gene mutation. And the Hereditary Spastic Paraplegia Foundation came to us about 10 years ago, and they said, "Oh, we've been talking to people working in STEM. Do you reckon you could find us a drug for our patients? It's rare, it's 5 in 100,000 people." And I said, "Oh, I'll give it a crack. I reckon we might be able to. This is what we do." And I showed him that slide of that circle of activity that I just showed you. And we took patient cells, we took healthy control cells. And for one reason or another, see, what you can do is you look at the biology and you focus in on some kind of assay that you might use that is showing the difference in biology, and you can use that assay to screen for drugs which treat that biology, right? That's the approach. And it really doesn't matter what that biology is as far as you're concerned, but you can see on the left there are these little green dots which are moving a lot more than the one on the right. Can you see that? So you can even eyeball it, but of course, being scientists, we could look at this with computer imaging and you know, image hundreds of these little green dots. And these little green dots happen to be this particular structure inside cells, which are very important for their function. And we show that these patient cells have slow, they're called peroxisomes, and there's part of their cell function is regulating what we call oxidative stress. So that's biochemical changes that happen just through activity inside cells, and cells have to regulate this, have to keep it low, and if they don't, they die. And we found that the cells were dying. And then we looked and thought about the biology, and we've got drugs that turn those ones in the patient speeds to the same as the controls. And that reversed the oxidative stress problem, stopped the cell death. So, and there are a number of different drugs, and we've gone for some drugs that actually have been used in cancer research. If you give lots of it, you'll kill the cell. But if you just give a little bit, you can make this reversal. And the good thing about these drugs is they've already been used clinically. So we know what their safety profile is, and we know they can get into the brain and the spinal cord where the diseased cells are. So that's our approach. We can use these drugs that have been used, we have a safety profile, and we're now taking them into a clinical trial. The first time there's a treatment like this for this disease. So that shows us that we can take these cells and take this kind of cancer biology approach to find drugs and treat disease. And so let's get back to Parkinson's disease. So we've got these, in that orange box there, we've got we know what a whole lot of genetic risks are, but most people don't have a genetic risk identified, but they must have some kind of unidentified genetic risks, otherwise they wouldn't have Parkinson's disease. Now, we know there are environmental factors that act. And for example, and this is kind of work that George Mellick has done in the past, whom many of you might know. George is in our team at Griffith and has been leading the Parkinson's team. That people who are born in the country have a higher risk of Parkinson's disease. And you may know that. Why is that? People have drilled down on that and found that if you have exposure to agricultural chemicals, you've got a higher risk. And that's been drilled down further to particular chemicals, a couple of which are rotenone and paraquat. So those are risk chemicals. And the question is, how do they act on cells? So we know they do act on cells, and they act on cells like rotenone, for example, increases oxidative stress by a different pathway, a different cellular pathway than I was describing for hereditary spastic paraplegia. But ultimately, once you get down to that level, the cells are going to die if you can't regulate that oxidative stress. And we know that the genes, the familial rare gene mutations, when people have drilled down there and said, well, what does that gene do? What does that gene do? They all are part of this oxidative stress and central in this oxidative stress regulation. So the concept is that environmental factors and gene factors are acting through common pathways to cause death of those neurons in the brain. And they will be also affecting other cells of the body, but not so dramatically. So we wanted to say, well, look, if you have Parkinson's disease, are your cells more sensitive to rotenone, this environmental risk factor? Because epidemiology doesn't prove anything, it just shows you the association between, let's say, rotenone and disease risk, but doesn't show cause. To show cause, we have to show actual effects in cells, and that's exactly what we've shown. Now, I apologise for your eyesight, especially up the back and the size of this, but I hope you can see a little bit of science in these graphs. And if you look, let's say the top left-hand corner, you'll see a line that's defined by the black dots and a line that's defined by the white dots. The x-axis, that's the numbers running along the bottom of each of these graphs, is time to 5 days. And the y-axis, that's the left-hand graph, the left-hand axis, shows you cell survival. One is 100% and it goes down to 40%. Now, these cells are exposed at, on the left-hand side, it's time 0, to rotenone. And the black dot lines are when you do that with healthy control cells. And you'll see that over those 5 days, exposed to that rotenone, about 90% are surviving, whereas the patient cells, it's gone down to 80%. So that tells us that this particular dose of rotenone, this particular concentration of rotenone, the patient cells are more sensitive to rotenone. That's important because that says they are people with Parkinson's disease who have some genetic susceptibility which we don't know, but all of them are more sensitive to this environmental risk factor. So the top left was our first study done with 8 patients and 8 controls. Well, as scientists, we want to repeat that. So we took 19 patients and 19 controls separately because we have this big neurobank of cells. And you can see the graphs are pretty much exactly the same. So that means that we can be confident that the patient cells really are more sensitive to this environmental risk factor. And on the right-hand side, there are a couple of other chemical exposures which show that they're also causing oxidative stress, and you can also see the patient cells are more sensitive than the controls to those other kinds of drugs or chemical exposures which also could come from the environment. The other question we want to ask is this specific to people with Parkinson's disease? If we take people with any neurodegenerative disease, will they also have this? Now that's important for a biologist because we're saying that the Parkinson's disease genetics points us to these particular cellular pathways, and so does the environmental exposure of rotenone. But other diseases which we know have different genetics, are they going to show the same thing? Well, they don't. So the top left-hand corner, you can see those two graphs are overlaying themselves, and they're comparing patients with motor neuron disease versus controls. And the next one on the left down is hereditary spastic paraplegia. We know they're under oxidative stress for different reasons, but here they're not sensitive to rotenone. So that gives us confidence that we're looking at something, this biology is worth pursuing in people with Parkinson's disease. Then on the top right, George, through his genetic studies, has identified some patients with some of these higher risk gene mutations for Parkinson's disease, shown there in red. But you can see those very strongly genetic forms of Parkinson's disease, those cells behave in the same way as the population of cells for whom we have no genetic information. And then the bottom graph kind of plots these all over together, showing that the genetic risk, the high genetic risk patients basically overlay the population, we call it idiopathic, which just means if a doctor tells you you've got idiopathic disease, it just says, we don't know why it's caused. That's their jargon for who the hell knows. Anyway, but the important thing is that we're showing this sensitivity to this environmental risk factor in cells with people with Parkinson's disease. And we could use that now as an assay to look for drugs. And we, at our university, Griffith University, we're very fortunate that our original director, many years ago, starting in 1973, started a collaboration with that big drug company, AstraZeneca. It's now AstraZeneca, back then Astra Pharmaceuticals. Over 10 years, his contract amounted to $100 million to isolate, to grind up every plant in Queensland because of its biological diversity, and pretty much everything that was squishy on the Great Barrier Reef, and extract drug-like compounds, drug-like chemicals from all of those. And they had the Queensland Museum out there looking for plants, and they had the Australian Institute for Marine Science looking at the Great Barrier Reef. They've identified multiple new plant species, multiple new sponge and coral species, and we have left with us now in our institute all of those samples ground up. They just look like brown powder to start with, all of them with a barcode, all of them with a GPS coordinate, and we have 45,000 of these, and from these we've extracted these drug-like compounds, 200,000 of them. Now, still, about 50% of the drugs we take are based upon plant derivatives. Originally, folk remedies. This worked, why don't we try that? Let's extract out the active compound. So this is still a vital source of our new drugs. And our approach is this, that we find some assay in the cells, and here we have cells growing, it doesn't matter what, but we can measure a fluorescence, we can measure a colour change, we can measure something happening in those cells. And we do our experiments, you can't quite see this, you don't do the experiment one by one, you do this now in plates that have 96 wells, so you can do almost 100 assays at the same time. Well, actually now, you can go up to 1,536 at the same time, which you need that kind of level of industrial biology if you're going to screen hundreds of thousands of chemical compounds. And also at Griffith University, we now house Australia's chemical library. So all the chemists around Australia and all the big institutes around Australia who've got chemists, they put their chemical compounds in our automated, computer-operated chemical library. We now have 600,000 compounds, pure chemicals, plus our 200,000 plant-derived chemicals. And this has robots, of course, to go and select out our samples, take minute amounts of those, put them out in these plates, and we send them nationally and internationally to biologists who are looking for drugs for brain diseases as we are, or for malaria, or for tuberculosis, or all sorts of diseases. And they go out to people around the world because everyone needs new chemical structures to investigate for drugs. So we have, to give you the example, our first screening, and we haven't done this for a while, but we came down from, we screened 40,000 compounds, and we've ended up with 4 drug leads. And the assay was, we know rotenone is specific for having an effect on our cells. The assay was, if we put in rotenone, we look for drugs that reverse the effect of rotenone. So we're looking at a biology that's relevant to people with Parkinson's disease specifically, and we're looking, it's relevant both to the genetics and also to the environmental exposures. So we have 4 chemicals now, which are at the beginning of that drug development. We know they are active, they have a biology that's important for Parkinson's disease, and now we want to take them through further to explore for Parkinson's disease. And this has got potential because we've only looked at 40,000 compounds, or we've potentially got 1,000 compounds, I mean 1 million compounds, 800,000 compounds, which we could explore to find other active drugs. And that's really a hope for the future and future funding. Oh, and this is just, I should go back and thank my collaborators. This work, Carolyn and I actually started the Parkinson's project. Carolyn is a neurologist at the Royal North Shore Hospital in Sydney, and we started the Neurobank with Parkinson's patients, of her patients. Subsequently, we're working on that, and subsequently we started working on hereditary spastic paraplegia. And Gautam's a PhD student of mine and now works with Carolyn in Sydney, and Fan is a young postdoc researcher who worked on this as well and was a major collaborator there. And Parkinson's project, as I said, is now led at Griffith by George, and Stephen is a major cell biologist who's part of that collaboration. Now, just to finish up, I've been talking about the commercial imperative. I've been talking about mesenchymal stem cells. I just thought this was such a great photo. I can't remember which museum I found that in. But because bone marrow there's now a lot of regulation about taking out cells from people who transplanted them, and rightly so. But bone marrow transplants have been grandmothered in, because basically all you do is you take some blood, you centrifuge it, you suck off the top layer of white cells and put them back in. So you're not manipulating the cells much, they're not going to change much through that process, and you're putting them back into the same person, so there's no immune problems. Well, since you can get mesenchymal stem cells from fat, lots of people, and there's bubbling around there in the scientific literature that mesenchymal stem cells can have some effects in some diseases. They're usually mild, and they're usually an anti-inflammatory effect, but they are there. So anybody who can suck out fat, and I'm not kidding, plastic surgeons who are sucking out fat set up stem cell clinics, because all you have to do is you spit it down and you take off the cells on the top, just like with bone marrow cells, and you can inject them back into the patient, no immune problems. And so then they fly under the regulation of the stem cells that would be transplanted into people with Parkinson's disease, which get grown in the dish. They have to be highly regulated, have a product, it's got to be specified, it's got to be regulated before it goes back in. So you can pay a lot for these unproven treatments. And that's, it's stimulated by all of this, all the press, which these are headlines from over the years of hints that we get from various studies, usually mice looking at mouse stem cells. So there's a lot of hype. And when you go to Google and say stem cell treatments, these are just some of the websites that you get, very slick. They come from all over the world. I think there's 16 places in Australia that will do this for you. You think, oh, well, we're in Australia, this only happens in Russia or Mexico or India. No, it happens all over the world, and in pretty much every country of the world. But it's fed by hope, isn't it? We all want to do whatever we can, but it's much better to spend your money on your current life than it is to give it to somebody else to spend on their life. So Dr. Google should be warned against, but if you do go to Google and you're looking for Parkinson's disease, you probably know, try to go to the university one, or the Cancer Council of Australia, what its equivalent for Parkinson's disease, or the NIH, or look at the address line and don't believe just because somebody's got a nice web page they're going to have nice things for you. And this is just an illustration. Since the early 2000s, now we have this plethora of journals, all with stem cells in the title. And they're all representing publications that are coming out, maybe 16 a month, maybe 16 a week. There's a huge, huge stem cell biology industry now because it's excited scientists, and the good thing for us is that means there's going to be progress in biology and progress in treatments. And I hope I've illustrated to you that even when you get this progress, you can see some of the many steps you have to get to, as I tried to take you through that drug discovery process. It's a long process. And also, most of our labs, you just have a couple of people working on it. In hereditary spastic paraplegia, most of that work was just done by those two people, Gautam and Fan, and over 10 years. If you had a lab where 100 people were doing this, you could do lots of things in parallel and accelerate it. And that's the kind of work that drug companies do, isn't it? But all the drug companies, all the biggest drug companies, you'll be frightened to know, have pulled out of their neuroscience research because they've spent hundreds of billions of dollars doing the biology, doing clinical trials, and not finding any drugs that work for brain diseases. 100 drugs have gone to phase 3, that's when you're getting them out into the population in big trials, in motor neuron disease, and a similar number in Alzheimer's disease. None of them worked. So you have to ask what's going wrong. And part of that is because the way they've investigated, what we call the models, they've used Alzheimer's disease. These drugs cure motor neuron disease or Alzheimer's disease in their mice, but mice are not humans. So I'm a great flag waver for looking at human cells in the hope that this will give us a new direction, and human stem cells in their different forms, patient and controls, to give us new ways, because now they come from humans, and we're sure to find out much more about the disease than in the mouse. So the take-home messages are then that cell transplantation is on the way, new drugs are on the way, but it's a slow process. But we hold hope. Thank you very much.
Alan has kindly will take some questions. Trevor, would you like to carry the torch? Can people please bear with us? We'll have to do something about the amplification on this, but I think it's better than nothing. So the question will be loud and clear. The answer might not be.
I'll speak more loudly.
Testing, yes. Hi, I'm Madonna. I'm from the Young at Park support group. We're in Brisbane. It's wonderful to hear the hope in your voice, and it's great to have you here today and all the people who are interested. You touched on the big pharmaceutical companies have pulled their money. I heard that Pfizer had just pulled theirs, but when a giant as big as Pfizer takes their money out of new drugs, it makes us wonder what is going on there. Can you shed any light towards that situation?
The reason they pulled out is, as I've said, how's that for up the back? So the reason for pulling out is because they're not making any money and they're spending a lot of money. The assumption, and it's not, Pfizer's just one of the recent ones. If you look at AstraZeneca, Merck, Pfizer, Novartis, they're closing down research labs that have 300, 600 people closed. So some of them are putting back collaborations in universities. And the others are just relying on, I think, universities, people like us, to come up with new things. And when they've come up with new things, then the drug companies, of course, if they see a winner, they'll jump on it. But at the moment, they're just very, very cautious, and they're moving to cancer research because that's where there are lots of people. If you find a drug for a common disease, then which has to be taken all the time, you make more money. So they have the commercial imperatives there for them to make money, obviously. And I'm just assuming that when the publicly funded and philanthropically funded research like ours comes up with something, then the big companies will come back. But just nothing is working, and it's because I think the history of this kind of neuroscience research is that you go to people with diseases, families with diseases, like some of these families with these high-risk genes. you identify the gene, and then you genetically modify a mouse, and you ask what effects that has on the cells. But the fact is that the cell regulation in a mouse is not the same, that there are lots of other genes that contribute that are not in a mouse. You know, mice are not humans. And they're finding that out in a big, big way and pulling out.
Just a question that maybe you can answer is about neuroplasticity. And can you see evidence of neuroplasticity as being a real thing?
Neuroplasticity is certainly very trendy out there in the world. The brain is a great organ for plasticity, for just learning is a part of that plasticity, but also relearning after you have a stroke. You know, that part of the brain is damaged, but you can relearn, and other parts of the brain will take over. In Parkinson's disease, let's say for the loss of these dopaminergic cells, there's nothing to take over. They're driving the part of the brain that's making decisions about movement. So the only way we can do that is to get more cells of those particular type in. The plasticity in the brain is much more likely to be found up in the cortex, let's say the thinking part of the brain, where it can reorganise itself much more easily. And the cells all have similar connections and similar structures, and so they just pass their functions over to the other cells. But in this loss of dopamine cells, it's a little focused area that just supplies the dopamine. The neuroplasticity will help people cope, but it won't treat.
Ray Fairfield from Toowoomba. Can you suggest any way that volunteers from groups like us can help your research?
Well, I think that, you know, if we need to make any more biopsies and get more patients into our neurobank, that's one way that people, if they volunteer and they're healthy relatives or healthy spouses, we're always appreciative of that. We're not actually recruiting at the moment, but that's one way. The other would be to be a bit more, venal, shall we say, is to help us in our philanthropic fundraising activities to get this up and running. We've kind of, the Parkinson's disease and other of our neurobank work has dropped down because of lack of funding in the last few years, and so I'm really at the moment, now that I have this platform, I'm now in a position where I'm doing a lot of fundraising to try and get this up to really serious levels of money where we can really accelerate the process of drug discovery for Parkinson's disease.